Hello everyone! We will study embryology. As we all know, animals begin life as a single cell. That cell must produce new cells and form increasingly complex structures in an organized and controlled manner to reliably and successfully build a new organism. As an adult human may be made up of around 100 trillion cells. This must be an impressively well-choreographed compendium of processes. What is embryology? Embryology is a branch of biology that studies the early formation and development of the organisms, from fertilized ovum to newborn individual. The research contents include the formation of germ cells, fertilization, embryo development, and the relationship between the embryo and the mother, congenital malformation, etc. It takes 38 weeks from fertilization to parturition of a mature fetus. It can be divided into 3 periods. 1. From fertilization to the second weekend, the bilaminar embryonic disc appears as pre-embryonic period. From the 3rd week to 8th weekend, the embryonic period, the preliminary formation of embryonic organs, systems, and shape. From the 9th week to the birth, the fetus gradually grows up. In this period, the organs, systems continue to develop and take shape, and some organs have certain functional activities. It can also be simply divided into 2 periods. Embryonic period is from the fertilization to 8th week. At the end of the period, the embryo has begun to take human shape. Fetal period is from 9th week to birth. The fetus grows. The organs, systems continue to develop. The picture above shows the formation of fertilized ovum, cleavage, 2-cell, 4-cell, 8-cell stages, morula, blastocyst, and implantation. The following picture shows the change of fetal shape from the 9th to the 38th week. Embryology includes the following branches. Descriptive embryology, which means using the methods of histology and anatomy, such as light microscopy and electron microscopy. The basic content is to observe the morphological evolution process of embryo development, including the evolution of shape, the evolution from primitive organ to permanent organ, the formation of system, the proliferation, migration, and apoptosis of cells, etc. Comparative embryology. It is to compare the embryonic development of different species of animals, including humans, in order to provide a basis for the study of biological evolution and its internal relationship, while it's conducive to deeper understanding of the development of human embryos. Experimental embryology, using the stimulation of chemical or physical factors, or the application of forced microsurgery, such as embryo cutting, cell transplantation, in vitro culture, etc., is applied to the embryo or the embryo tissue cultured in vitro, so as to observe its influence on the embryo development and to study its internal and mechanism. chemical embryology. With the development of chemistry, biochemistry, and histology, people use these techniques to study the changes of some chemical substances in the cells and tissues and the chemical basis of morphogenesis during embryogenesis. Molecular embryology, to study the molecular basis of biological processes such as fertilization, implantation, cell differentiation, tissue induction, and cell migration, with the theory and technology of molecular biology, to explore the time sequence, spatial distribution, and the regulatory factors of gene expression in the process of embryogenesis, to study the role of gene expression products, that is, various proteins in embryonic development, to clarify the molecular process and mechanism. Researchers often use gene knockout and transgenic technology to obtain model animals with different phenotypes, which creates conditions for the study of gene function and the establishment of disease model animals. Reproductive engineering, through artificial intervention in the early reproductive process, in order to obtain the desired new individuals. The main techniques are in vitro fertilization, early embryo culture, embryo transfer, intracytoplasmic sperm or nuclear injection, gamete and embryo cryopreservation. Look at this picture. Test tube babies and cloned animals are the most famous achievements. Do you have this question? Why study embryology? Our biological development is a fascinating subject, deserving study for interest's sake alone. Our understanding of embryological development also helps us answer questions about our adult anatomy, why congenital abnormalities sometimes occur, and give us insights into where we come from. In medicine, the importance of an understanding of normal development quickly becomes clear as a student begins to make the same links between embryology, anatomy, physiology, and neonatal medicine. That's all. Thank you.